Hello, my name is Katrin Schöning-Stierand from the Center for Bioinformatics at Universität Hamburg in Germany. Today I will talk about GeoMine, a web-based tool for chemical three-dimensional searching of the PDB. At our institute, we host the Proteins Plus web service that provides different tools for the selection and preparation of protein structures in life science research. One of these tools is GeoMine, that gives insights in the geometric arrangement of protein-ligand interfaces and enables textual, numerical, and chemical 3D searching in the PDB. There are many geometry-related questions in the context of protein-ligand interfaces like, is this a reasonable geometry for a hydrogen bond? Or, what is the typical distance between two interacting atoms? Or, is there any preferred residue for the interaction with a specific functional group? Or, which functional groups can be found in similar protein environments? GeoMine uses a technology that has been developed at our group a few years ago and was implemented at first in the desktop tool Pelican. Pelican is a database that contains data derived from the PDB structures of protein-ligand complexes. Previous to the database construction, the structures are preprocessed with Naomi. That means, for example, small molecules are extracted, binding pockets are determined, correct tautomeric forms are assigned, and hydrogens added. For the complex, many textual and numerical properties are then stored, like the resolution and PDB title, the EC number of the protein, properties of the pockets, and physicochemical properties of the ligand. Every heavy atom in the pocket is a potential search point for a query and is stored together with a list of properties like its element, the residue or functional group it belongs to, or the interaction type. Interactions are pre-calculated for each pocket and also stored in the database. A tailor-made index enables query times of all these data in the range of seconds to a few minutes. Constructing a query starts with the definition of points, which can either be atoms or ring centers. These points can be further defined by different attributes. For example, the interaction type can be specified, or for ligand points a functional group, and for protein points an amino acid can be chosen. Additionally, the environment of each atom can be defined by means of a smarts. In the next step, point-point constraints can be defined using a distance range or a pre-calculated interaction type. Then angle constraints can be added to the query. An angle can be defined between any pair of point-point constraints or between points with an interaction direction. A ring point, for example, always has its ring normal as direction. As a last step, the query can be combined with any property constraint for properties that have been stored in the database. After the query has been set up, all matches within the database are searched. If you are interested in the details of the query mechanism and the user interface for the query construction, please visit our poster and listen to the poster talk, which are both presented today and tomorrow in the poster session. As already mentioned, Pelican is a desktop application and consists of three parts: the Pelican database, a graphical user interface that supports the user in the query construction and the inspection of results. The database and the graphical user interface are connected by the tool. Our idea was to make this technology available as a web service for all life scientists, and in this way to negotiate the big installation hurdle of a local desktop application. So we developed GeoMine based on the Pelican tool, and this is how it looks now. The graphical user interface is integrated in the openly accessible Proteins Plus web server that already provided a 3D panel for molecule visualization. In order to enable a high query versatility and precision, but also a reasonably short runtime, the algorithm has been refined in several ways. Compared to Pelican, improvements of the search process and the indexing method reduced the runtime notably. In addition, the switch from an SQLite database to a server-based PostgreSQL database leads to a better memory management, scalability, SQL compliance, extensibility, and multi-user handling. On the following slides, I will show an application example. The presented use case is an off-target search for celecoxib, which is an inhibitor of the enzyme COX-2. The aryl sulfonamide moiety interacts via 3 hydrogen bonds with a protein. The shown query includes the interacting atoms of celecoxib and COX-2 and the hydrogen bonds in between. Additionally, the sulfon atom and the center of the phenyl ring are part of the query. This query is an example for being constructed as chemically quite precise and geometrically rather vague. This slide shows the user interface for the query construction. The selection of points and their connections can be done directly in the 3D visualization panel. All selected points are color-coded and correspond to one line in the table on the right-hand side. In the table, the points can be further specified with different properties. GeoMine found 170 results in 60 pockets of 43 different PDB structures. The query processing on the database that contains at the moment about 350,000 pockets was performed in 43 seconds. The first 150 results are presented in the table on the right-hand side and can be toggled in the 3D viewer, where the structures are shown superposed to the user-defined query. Among the first hits was acetazolamide bound to carboanhydrase. And in fact, it is known from literature that carboanhydrase is an off-target for celecoxib. This picture shows celecoxib bound to carboanhydrase. Summing up, GeoMine enables three-dimensional searching in the PDB. The search infrastructure is freely available, user-friendly and web-based. For further use cases and complementary information on the technology, please visit our poster. Thank you for your attention.